scNovel. It is a scalable deep learning-based network for the novel real cell discovery in the single-cell transcriptomics. So for the pharmaceutical research and development pipeline here, it stands for the process for identifying a potential beneficial drug, proving that it is safe and effective, and making it available in a way that maximizes its benefit to as many patients as possible. And there are hundreds of millions of people have lived longer and a healthier life because of vaccination and other preventative measurements measurements and treatment and cures and so on. And for my research, I'm primarily focused on the first two steps, the target identification and target validation. Today, I'm mainly focused. I will mainly talk about my work involves in the first stage, the target identification, which involves the genetic disease association or analysis between the disease states and healthy states, and also some pathway or network analysis. This first step, target identification in the drug discovery process, is highly imperative to select the key targets relevant to a disease states and identify their ability to be modulated by a drug, thus hoping to increase the probability of success in the further clinical trials and drug discovery process. And in this step, many high-resolution sequencing techniques, like, for example, single-cell RNA sequencing and related computational tools have sprung up. The typical single-cell analysis pipeline here usually include the pre-processing and downstream analysis, and the downstream analysis can be cell or the gene level. Quality control, normalization, data correction, feature selection, and also dimension reduction are quite commonly seen in the pre-processing step. Also, for the downstream analysis, it usually involves aspects like cluster or cell annotations, trajectory, inference, differential expression, gene analysis, and so on. And for the fundamental and essential step in the downstream analysis should be the clustering or the cluster or cell annotation of the single-cell RNA sequencing data. This step, in this process, it is common to discover some novel or even rare cell type that were previously unknown or underrepresented in the existing knowledge. And I guess this is one of the exciting aspects of the single cell analysis, as it also allows us to uncover some previously uncharacterized cellular heterogeneity, and also can explore our understanding of the new cell populations. And the identification of novel cell types can also provide new insight into the molecular mechanisms underlying disease pathologies, which can in turn inform the drug target discovery efforts. And this is its relationship with the whole drug target discovery pipeline. And also, during the disease transitional states, we may also have some more critical rare novel cell types sprung up. There are some existing work about designed to detect the novel cells, such as scMap, scMPy, scPredict, and scLearn. They are all good tools. However, limitation lies in that first, they mainly focus on the single cell type annotation, but none of them is designed for the novel cell detection, especially for the novel rare cell detection. And also the previous work mainly employed the simple method like principal component analysis for feature extraction. The limitation here is that PCA will keep the majority cells feature, but usually ignore some weak real cells feature. The question is, do we really need a dedicated tool for the novel real cell type detection? To answer this question, we conduct extensive experiments and used AUROC value as our main evaluation matrix, and also incorporating the FPR95 AUPR for additional reference, since AUROC is more suitable for this setting when the classes are in balance. FPR95 can be represented as the probability that a negative sample is misclassified as positive when the true positive rate is as high as 95%. And in this table, we can find that though scMap, scPredict, and scLearn are all nice tools, but since they did not design for this task, for the novel real cell type discovering task, their performance is not good enough. All their AUROC values are below 90%. So we proposed a scalable deep learning based network especially for the novel real cell discovery called scNovel, and this work is recently accepted by briefings in bioinformatics and is now available online. Let's first look into the framework design of scNovel. The basic architecture of our proposed scNovel is a deep neural network-based classifier. scNovel takes the input of single-cell expression data and output the novel cell detection results based on the classifier's cell type probability and the predefined threshold. There are two stages of using scNovel. The first is training stage, and the second is query stage. Our special design for the novel real cell type detection task, in reflection to the module design, is we incorporate the barcode pre-processing module, and also we divide the query stage to the parallel query stage and the sequential query stage with different combination of core module and barcode pre-processing module. I will talk a little bit more detail about this part later. So overall, we developed an integrated pipeline for the identification of novel real cell and also provide deeper analysis or downstream analysis like biomarker detection, differential expression analysis, and also the gene set enrichment analysis. And our proposed scNovel-based analytical framework can be tailored to investigate genes associated with various diseases. In the training stage, we only use the known cell types data for training as reference, and we employ the softmax layer to calculate the cell type probability, and we also use the coarse entropy loss and the backpropagation to update the network's weight. During the query stage, we employ the barcode preprocessing as I have mentioned earlier. This module is specially designed for the novel real cell type detection task. By using this module, we are trying to enlarge the margin between the typical cells and the novel cells. And hence, applying this module, the score of the typical cell types will change more, also including the increasing more or decreasing more, while the score of the novel cell types does not change too much. Therefore, the margin between the typical cells and the novel cells can be enlarged. And based on different combination ways of the barcode preprocessing and the core classification module, we can divide our query stage into the parallel query stage and the sequential query stage. And based on these two stages, to access the nullity of a given cell, we provide 4 modes or 4 score function of inference based on different scoring function of the composition. And auto scoring function is defined as the max cell type probability, and we can have the score S. And if the score S is larger than the threshold, the cell type is defined as typical, and otherwise it is novel. Oops, sorry. Okay, sorry. Oh, okay. This figure gives an overview of the 4 different scores used in each model, in each query mode. For example, let's take the first and the simplest scoring function, SMSP, as an example. This scoring function is a classic score function without the barcode preprocessing module, SLIN, and we found this most performance is lower than the rest. Therefore, showing that the necessity of our incorporation of the barcode processing module in this novel real cell detection task. And I'm not going to more technical details about the rest 3 score function here. And if you are interested, please refer to our manuscripts. Now let's move on to the experimental results of scNovel. Here, the table shows the average experiment results across different data sets of scNovel and the baseline models. The red bold and the blue bold are the best result and the second best result respectively. Here, scNovel represent the number of novel classes, and we can derive that scNovel reached the most performance across all evaluation matrix. And also, it is the only method whose average AUROC values are all above 94%. In our experiment, we set the top scNovel's most rare cell as novel for testing or query, and the other cells as typical for training or reference. And the box plot here shows that scNovel has a higher mean but smaller interquartile range of AUROCs than all the other methods. Also, the significance of these improvements are annotated using the star marks. Overall, scNovel's AUROC value is up to 16.26% higher than the second best method, scNlearn on average. When the number of the novel class, which we denoted as scNovel, is set to 2, scNovel has the best novel real cell detection performance on the Zillions dataset. So in this dataset, PDC and RBC, these 2 cell types are said to be novel. The reason why we want to highlight PDC is because first, it is rare, because it constitutes less than 0.4% of all the PBMC cells. So it is definitely difficult to detect. And also, PDC have been shown to be important in the antiviral immunity and has also been implicated in the initiation and the developmental of many autoimmune and inflammatory diseases. The UMAP visualization here shows that almost all the models can successfully detect RBC. However, RBC here, it lies in the lower left region in the UMAP visualization. And however, only our map model, scNovel, can successfully detect the PDC cells, which is a novel real cell in this setting. So we show that scNovel has a robust performance on the real cell types prediction task. In the real application scenario, training and applying such real cell detection tool can be easily conducted on different datasets, different protocols, or even different platforms with heavily batching effects. And these semantic variations are crucial, particularly when analyzing the real cell population. And therefore, this heavy batch effect must be carefully addressed to ensure that the signal is actually accurately captured to produce an accurate and reliable result of the prediction. So we next move on to explore whether scNovel can generalize well to the cross dataset annotation. The result here shows that scNovel achieves the best performance on the intro dataset with figure A used the cell-seq as reference, while figure B used the PBMC 10x v3 as reference. And we can find that scNovel outperformed the SCMaps cluster by 7.31 and outperform scLearn by 9.13. In real-world single-cell analysis, computational efficiency is definitely important and can be viewed as the main obstacles to achieve the scalability of a model. From this figure, we can derive that the running time of scNovel in the red line remains relatively constant across all the datasets with varying sample numbers or the sample size. And also, scNovel is faster comparing to other methods, especially when the dataset is large. The reason behind this is that scNovel can directly handle the raw input without additional transformation. To further validate the robustness of our performance on the large and the imbalanced dataset, we apply scNovel on a million-scale COVID-19 immune atlas. And this dataset consists more than 1.5 million samples, and in total have 64 cell types. We can see in the figure B, the healthy group only has 54 cell types. Thus, there are 10 remaining cell types only exist in the novel groups, in the COVID group. So in this setting, we used all the healthy case cells for training, which contains 54 cell types. For the query stage, we put all the COVID cell samples into query, and only the 10 cells that only exist in the COVID case, but disappear in the healthy state are said to be novel. Figure C here gives a combinatorial view of the relationship between the cell number and scNovel's performance. Take the mass cell type and EPI82 cell type as example. These two cell types are quite rare, highlighted in the red box. We can see that the mass cell types only contain 17 cells, which is definitely a very rare cell types. But it achieves quite high performance on these two cell types in comparison with even the majority cell types. So therefore, scNovel shows great performance on the novel rare cell type detection, and even better than the novel majority cell type detection. Therefore, this success shows that our tools is especially dedicated for the novel rare cell type detection. Figure D here give a more comprehensive view of the four inference mode or the four score function that we have mentioned earlier. The relative lower performance of SMSP suggests that the importance of incorporating the AUROC preprocessing model in our framework. And overall, we can claim that scNovel is capable of accurately identifying the 10 novel rare cell types that are exclusively presented in the COVID-19 cases within the dataset comprising the millions of cells. Figure E and F are the UMAP visualization. You can see that the 10 cell types, novel cell types we detected are quite rare in comparison to the whole atlas cells. Studies have shown the importance of cell type specific analysis of the genetic heterogeneity of specific cell type in the field of biomedicine. And for instance, the COVID single cell atlas that we have used before contains, they detect 64 cell types and further analysis the relationship between all the cells gene expression and disease in their work. And however, the detection process of the novel cell types is quite time consuming and needs many prior knowledge. But our proposed scNovel could find such novel rare cell type automatically, which is definitely a good aspect. We then want to discover whether scNovel can discover or detect new or missed cell types. We adopt this million scale COVID atlas as reference and query on another COVID dataset. In our setting, the B cells and T cells are presented in the reference set, while 81 and 82 are left for testing. And from the results shown in figure C and D, we can find that B cell and T cell are less likely to be predicted to be novel in the result of scNovel, but 81 and 82 are successfully detected as novel cell type. So in this setting, we show that scNovel can successfully detect the novel cell type. And then we move on to, in total, find 23 cell types that does not appear in the million scale COVID atlas. From figure E, we can see that the gene expression pattern of all the 23 novel detected cell are different from the cell in the reference set. From the clustering location in figure C and D, and the high expression level of the CD 163 of cluster 3, 5, and 12, we may think that they can be the subtypes of macrophage. We then conduct deeper analysis of these 3 selected clusters. We conduct analysis like biomarker detection, differential expression gene analysis, and GSEA analysis of these 3 clusters. We want to study their relationship between their expression and the COVID-19. We investigate the changes in the biological pathway between the macrophage with differing COVID status and find a significant upregulation in the COVID samples, particularly in the neutrophy activation involved in the immune response. And this findings suggest the potential of scNovel as a critical tool in accelerating diagnostic and response in future pandemics or diseases. I also want to highlight that though in our experiment, we mainly use a COVID-related data set for the case study, but scNovel's analytical framework can be easily applied to various other diseases. At last, please allow me to express my gratitude to my collaborators, Zheng Chuanyang from CUHK, Cheng Yuqi from Georgia Institute of Technology, Xuesong Wang from CUHK, and Hong Xinwei from NTU.